Hello everyone, let's today read chapter 4 of microbiology, general virology and overview of viral infections. So what are the general property of viruses? Viruses are smallest unicellular organism that are obligate intracellular. Viruses are the most primitive microorganism infecting the man. They differ from bacteria and other prokaryotes in many ways. Viruses differ from bacteria as they are obligate intracellular. They possess either DNA or RNA but never both. Filtrate. They are smaller than bacteria, can be passed through the bacterial filtrate. They cannot be grown on artificial cell-free media. They can be grown in experimental animals, embryonated eggs, or tissue culture. They multiply by a complex method, but not by the binary fission as seen in the bacteria. Viruses do not have proper cellular organization. They do not have cell wall or cell membrane or cellular organelles, including ribosomes. They lack the enzyme necessary for protein and nucleic acid synthesis. They are not susceptible to antibacterial antibodies. Morphology of virus. The entire virus particle is called as viron. It comprises of a nucleic acid, which is either DNA or RNA, surrounded by a protein coat called as capsid. Together it is known as nucleocapsid. Some virus also has an outer envelope. Nucleic acid. Virus has only one type of nucleic acid, either DNA or RNA, but never both. Accordingly, they are classified as DNA virus and RNA virus. The nucleic acid may be single or double stranded, circular or linear, segmented or unsegmented. Most DNA viruses possess double-stranded DNA, except prevoviruses, which has single-stranded DNA. DNA viruses possess single-stranded RNA, except reovirus, for example, rotavirus, which possess double-stranded RNA. Retroviruses possess two copies of single-stranded RNA. The nucleic acid of most viruses are unsegmented, except Boyan virus, influenza virus, rotavirus, arena virus, which has multiple segments of RNA. Capsid. Capsid is composed of a number of repeated protein subunits, polypeptides called capsomeres. Function of capsid includes it protects the nucleic acid core from the external environment, for example, nuclease. In non-enveloped viruses, it initiates the first step of viral replication by attaching to specific receptor on the host cells, thus facilitating the entry of virus. It is antigenic and specific for each virus. Depending upon the arrangement, depending upon the arrangement of capsomeres surrounding the nucleic acid, three types of symmetry are described: icosahedral or cubical symmetry. The capsomeres are arranged as if they lay on the faces of an icosahedron. Such viruses have a rigid structure. All DNA viruses except pox viruses and most of the RNA viruses have icosahedral symmetry. Figure 4.1a and c. So this is figure 4.1a which has protein part, lipid part, envelope, capsid, nucleic acid. And this is the figure C, which shows capsomere and nucleic acid and capsid. Second is helical symmetry. The capsomeres are coiled surrounding the nucleic acids as in the form of helix or spiral. Such viruses are often flexible. Example includes few RNA viruses such as myxoviruses, rhabdoviruses, filovirus, Bornea virus, etc. So, capsomere, nucleic acid, envelope protein and lipid, and nucleic acid, which is spiral and surrounded by capsomere. Third is complex symmetry. Pox virus do not have either of the above symmetry but possess a complex symmetry. Now let's see envelope. Certain viruses possess an envelope surrounding the nucleocapsid. Envelope is a lipoprotein in nature. The lipid part is derived from the host cell. Host cell membrane, and the protein part is virus coded, made up of subunits called pepelomeres, which projects as spikes on the surface of envelope. Some viruses may have more than one kind of pepelomeres. For example, influenza viruses possess hemagglutinin and neuraminidase pepelomeres. Envelope viruses are more susceptible to heat and lipid solvents like ether. Pepelomeres are antigenic. They can also bind to specific receptor on the host cell, facilitating the entry of virus. Most viruses are enveloped except non-enveloped DNA virus, parvovirus, adenovirus, and papovavirus. Non-enveloped RNA virus like picornavirus, reovirus, calcicivirus, hepatitis A virus, and hepatitis E virus. Size of viruses. Viruses are extremely small, vary from 20 to 400 nanometer in size. Smallest virus is parvovirus 20 nanometer and largest being the pox virus 400 nanometer. Because of the small size, virus can pass through bacterial filter and they cannot be visualized under light microscope. Shapes of viruses. Most of the animal viruses are roughly spherical with some exceptions. Shapes of viruses. Rabies virus bullet shaped, Ebola virus filamentous, pox virus brick shaped, adenovirus space wheel shaped, rotavirus wheel shaped, tobacco mosaic virus rod shaped. Rabies is bullet shaped, Ebola virus is filamentous, pox is brick shaped, adenovirus is space wheel shaped, rota is wheel shaped, and tobacco mosaic is rod shaped. Viruses differ from viroids, prions, and virusoids. The entire viral particle is called as viron, comprising of protein, capsid, cord, and nucleic acid. There are some incomplete viral particles such as viroid, prion, and virusoids. Viroids comprise of naked circular small single-stranded RNA without capsid. They are mostly restricted to plants. They depend upon the host enzyme for replication Hepatitis D virus in humans is similar to viroids. Prions consist of abnormal infectious protein molecules without nucleic acid. They are highly resistant to physical and chemical agents. Now let's see some of the viruses. Parvovirus 18 to 26 nanometer, adenovirus 70 to 90 nanometer, bacteriophage 28 to 100 nanometer, influenza virus 80 to 120 nanometer, herpes virus 120 to 200 nanometer, rhabdovirus 75 into 180 nanometer, smallpox virus 230 into 400 nanometer. Comparison of sizes of viruses with that of E. coli. So E. coli, which is gram-negative bacteria, is this pink structure, and all these viruses get into it. So it is 1 to 3 micrometer into 0.4 into 0.4 to 0.7 micrometer. Now let's continue with the prions. They are highly resistant to physical and chemical agent. They produce neurodegenerative conditions in brain in humans, having long incubation period in years, called prion disease. Nomenclature and classification. International Committee of Taxonomy of Viruses in 2000 has proposed a classification of the virus. Classification of virus. DNA viruses, nucleic acid is DNA. Herpes viri D, nucleic acid double-stranded linear DNA which has envelope, icosahedron symmetry 150 to 200 nanometer in size. Representative viruses are herpes simplex virus 1, herpes simplex virus 2, varicella zoster virus, Epstein-Barr virus, cytomegalovirus, human herpes virus 6, 7 and 8. Second is Hypnaviridae, double-stranded circular incomplete DNA which has envelope, icosahedron symmetry 40 to 48 nanometer in size. Hepatitis B virus. Parvoviridae, single-stranded DNA which is linear, envelope is absent, icosahedron symmetry 18 to 26 nanometer in size. Parvovirus B19. Papoviridae, double-stranded circular DNA, absence of envelope, icosahedron symmetry 45 to 55 nanometer in size. Human papilloma virus, JC virus and BK virus. Poxviridae, double-stranded linear DNA which has envelope, complex symmetry 230 into 400 nanometer in size. Variola smallpox, molluscum contagiosum virus. Adenoviridae, which has double-stranded linear DNA, envelope is absent, icosahedron symmetry 17 to 90 nanometer. Human adenovirus. Picornaviridae, RNA viruses which has RNA, single-stranded positive sense of RNA, absence of envelope, icosahedron symmetry 28 to 30 nanometer in size, which includes poliovirus, Coxsackie virus, ecovirus, enterovirus, rhinovirus, hepatitis A virus. Calciviridae, which has single-stranded positive sense RNA, absence of envelope, icosahedron symmetry, 27 to 40 nanometer in size. Norwalk virus, hepatitis E virus. Togaviridae, single-stranded positive sense RNA, which has the envelope icosahedron symmetry, 50 to 70 nanometer in size. Rubella virus, Eastern equine encephalitis virus, Western equine encephalitis virus. Flaviviridae, single-stranded positive sense RNA, which has envelope icosahedron symmetry, 40 to 60 nanometer in size. Yellow fever virus, dengue virus, Strenus Lewis encephalitis virus, West Nile virus, hepatitis C virus. Coronaviridae, single-stranded positive sense RNA, which has envelope helical, 120 to 160 nanometer in size. Coronavirus such as SARS-CoV, MERS-CoV, SARS-CoV-2, the agent of COVID-19. Rhabdoviridae, single-stranded negative sense, which has envelope helical, 75 into 180. Rabies virus, vesicular stomatitis virus. Filoviridae, single-stranded negative sense RNA, which has envelope helical symmetry, 80 into 1000 nanometer in size. Marburg virus and Ebola virus. Paramyxoviridae, single-stranded negative sense RNA, which has envelope helical symmetry, 150 to 300 nanometer in size. Parainfluenza virus, Mumps virus, measles virus, respiratory syncytial virus, Nipah virus, Hendra virus, Newcastle disease virus, metapneumovirus. Orthomyxoviridae, which is single-stranded negative sense RNA, 7 to 8 segments, which has envelope helical symmetry, 80 to 120 nanometer. Representative virus, influenza viruses A, B, C and D. Bunniaviridae, single-stranded negative sense RNA with 3 circular segments, which has envelope helical symmetry, 80 to 120 nanometer. Hantavirus, California encephalitis virus, sandfly fever virus. Arenaviridae, single-stranded negative sense, 2 circular segments, which has envelope helical symmetry, 50 to 300 nanometer in size. Lymphocytic choriomeningitic virus, Lassa fever virus, South American hemorrhagic fever virus. Reoviridae, double-stranded, 10 to 12 segments, absence of envelope, icosahedron symmetry, 60 to 80 nanometer in size. Rotavirus, reovirus, Colorado tick fever virus. Last one is Retroviridae, 2 identical copies of positive sense single-stranded linear RNA, which has envelope icosahedron spherical symmetry, 80 to 110 nanometer in size. HTLV, human T lymphocyte virus, and human immunodeficiency virus. SARS-CoV, severe acute respiratory syndrome coronavirus. MERS-CoV, Middle East respiratory syndrome coronavirus. SARS-CoV-2, severe acute respiratory syndrome coronavirus. Some families, Pox viridae, Herpes viridae, Parvoviridae, and Paramyxoviridae are subfamilies which in turn consists of genera. Viral replication. Viruses do not undergo binary fission seen in bacteria but undergo a complex way of cell division. Replication of viruses passes through 7 sequential steps: attachment, penetration, uncoating, biosynthesis, assembly, maturation, and release. This can be asked in Viva, so the order should be remembered correctly: attachment, penetration, uncoating, biosynthesis, assembly, maturation, and release. First, absorption or attachment. It is the first and most specific step of viral replication. It involves receptor interaction. The virus have attachment sites on their envelope or capsid protein that binds to the complementary receptor sites present on the host cell surface. HIV viral surface glycoprotein 120 binds to CD4 molecule on the host cell. So attachment, first and most specific step of viral replication. It involves receptor interactions. The viruses have attachment sites on their envelope or capsid proteins that binds to complementary receptor sites present on the host cell surface. HIV virus viral surface glycoprotein 120 binds to CD120 molecule of the host cell. Influenza viral hemagglutinin and envelope protein binds specifically to glycoprotein receptor present on the surface of respiratory epithelium. So what happens in absorption? This is the most specific step. This word should be remembered. So there occurs receptor interactions. So virus have attachment sites to the host on their envelope or capsid proteins if they lack the envelope that will bind to complementary receptors which are present on host cell. So what happens in HIV virus? The surface glycoprotein 120 binds to CD4 molecules of the host cell. And in case of influenza, viral hemagglutinins, which is an envelope protein, binds specifically to glycoprotein receptors present on the surface of respiratory epithelium. Penetration. After attachment, the virus particles penetrate in the host cell either by phagocytosis or viropexis. It occurs through the receptor-mediated endocytosis, resulting in the uptake of virus particle within the endosomes of the host cells. So how the virus penetrate the host cell? Via phagocytosis, which is also called as viropexis, where there occurs the receptor-mediated endocytosis. So the virus gets embedded into the host in the form of an endosome. Second is membrane fusion. Some enveloped virus For example, human immunodeficiency virus or HIV enter by fusion of their envelope protein with the plasma protein of the host cell, so that only the nucleocapsid enter into the cytoplasm, wherever the viral envelope remains attached to the host cell membrane. So what occurs in membrane fusion? In case of enveloped virus like HIV, there occurs the fusion of enveloped protein and plasma membrane of the host cell. So as they both fuse, so the matter which is present inside the envelope, that is nucleocapsid, enter into the cytoplasm of the host, whereas the viral envelope remain attached to the host cell membrane outside only. Then injection of nucleic acid. Bacteriophage, virus that infects the bacteria, cannot penetrate the rigid bacterial cell wall. Hence, only the nucleic acid is injected while the capsid remains attached to the cell wall. So we have seen two steps. First was attachment or absorption and second was penetration. Now let's see the uncoating. By the action of lysosomal enzyme of the host cell, the viral capsid gets separated from and the nucleic acid is released into the cytoplasm. This step is absent in bacteriophage. Viral capsid gets separated due to lysozyme from the nucleic acid. But in case of bacteriophage, there occurs injection of only the nucleic acid, so there is no need for the separation as capsid does not enter the the bacterial cell. So we have seen the three steps: attachment or absorption, penetration and uncoating. Now let's see biosynthesis. In this step, following the viral components are synthesized: nucleic acid, capsid protein, enzymes required for various stages of viral replication, regulatory protein to shut down the host cell metabolism. Site of nucleic acid replication. In DNA viruses, the DNA replication occurs in the nucleus, except in the pox virus, which synthesize the DNA in the cytoplasm. In RNA virus, the RNA replications occur in the cytoplasm, except the retrovirus and orthomyxovirus, which synthesize RNA in the nucleus. DNA viruses. Biosynthesis of DNA virus involves the following basic steps: transcription of parental DNA to form the early messenger RNA, mRNA. Early mRNA undergoes translation to produce early non-structural protein. Viral DNA replication. Early non-structural protein shut down the host metabolism and helps in the replication of parental DNA to form copies of progeny DNA. So for the biosynthesis of DNA virus, first there occurs the transcription of DNA to mRNA of the parental DNA. Then there occurs the translation of mRNA into protein to produce early non-structural protein. Then the non-structural protein will seize the host cell metabolism and will help in the replication of parental DNA to form copies of progeny DNA. Steps of DNA virus biosynthesis. Transcription for early genes, then translation parental DNA to early mRNAs to early proteins, then replication of viral DNA, parental DNA. There occurs transcription of late genes, late mRNAs, then translation to form late proteins. Assembly of nucleocapsids, daughter viroids. Steps of DNA virus biosynthesis. Progeny DNA viruses undergoes transcription to form late mRNA, which are further translated to form late structural proteins. Capsid and enzyme envelope viruses. Minor differences in replication. Minor differences in replication among DNA viruses. Site. DNA replications occur in nucleus except in pox virus, where it occurs in the cytoplasm, whereas mRNA transcription and protein translation takes place in the cytoplasm. The linear double-stranded DNA of herpes virus becomes circular inside the host cell and then replicates by rolling circles mechanism. Hepdina virus, hepatitis B virus contain partially double-stranded DNA that is first converted to fully double-stranded DNA by viral polymerase. RNA viruses. The process of biosynthesis varies among RNA viruses depending upon whether the genomic RNA is positive, negative sense, or single or double-stranded. First, positive sense single-stranded RNA virus. In most RNA virus, their RNA have the same polarity as mRNA. Hence, they are directly translated to form early proteins. Synthesis of progeny RNA. Early protein have RNA polymerase activity that direct replication, transcription. Positive single-stranded RNA forms negative single-stranded RNA to positive single-stranded RNA. Late protein are formed by translation of positive single-stranded RNA. So, in case of positive single-stranded RNA, there is direct translation to form early proteins. Late proteins are formed by translation of positive single-stranded RNA. Type 2 negative sense single-strand RNA. Myxovirus and rabies virus have negative sense single-stranded RNA. Their RNA polarity is opposite to that of mRNA. Hence, they cannot directly translate into the proteins. Negative single-strand RNA transcribes into positive single-strand RNA first using viral RNA polymerase. Then the positive single-strand RNA translates to form protein, and also it acts as a template and undergoes replication to form copies of negative single-stranded RNA. Type 3 double-stranded RNA viruses. Reovirus, for example, rotavirus, have double-stranded RNA, which is usually segmented, with each segment coding for one polymerase. The positive strand RNA can act as mRNA and undergoes translation to form protein. by viral enzymes. Both positive and negative strand RNA serve as a template for synthesis of complementary strands to form the duplex. Translation: positive single-stranded RNA forms early viral proteins, acts on RNA polymerase and undergoes transcription to form negative single-stranded RNA, then further transcription to form positive single-stranded RNA, then translation, late viral protein assembly. Then negative single-strand RNA undergoes transcription, viral RNA polymerase, positive single-stranded RNA translation, viral proteins serve as mRNA, transcription, viral RNA polymerase, the negative single-stranded RNA gets assembled in the daughter virons, then double-stranded RNA segment, viral RNA polymerase, negative RNA and positive RNA translation serve as mRNA and viral proteins are synthesized. Transcription, viral RNA polymerase, positive RNA and negative RNA, double-stranded RNA assembly to form the daughter cells. Fourth, type 4 retrovirus HIV and other retrovirus possess two copies of linear non-segmented positive single-stranded RNA and enzymes such as reverse transcriptase and integrase. After the entry in the host cell, the single-stranded RNA gets reverse transcribed to form single-stranded RNA by viral reverse transcriptase, which acts as RNA-dependent DNA polymerase. DNA is to RNA hybrid is formed. Reverse transcriptase has also ribonuclease activity by which it digests the RNA from DNA is to RNA hybrid. The resulting single-stranded DNA is converted to double-stranded DNA by the DNA polymerase activity of the same reverse transcriptase enzyme. Double-stranded DNA is transported into the nucleic acid where it gets integrated into the host chromosome by viral integrase. The integrated DNA serves as a template for production of mRNA, which can be translated into proteins and genomic progeny. Fifth is the assembly. The viral nucleic acid and proteins are packaged together to form the progeny virus nucleocapsids. Assembly may take the place in the host cell nucleus or cytoplasm. DNA virus are assembled in the nucleus except the herpesvirus and poxvirus in the cytoplasm. RNA virus are assembled in the cytoplasm. So in the assembly, nucleic acids and proteins are packaged together to form the progeny virus, which is nucleocapsid. So this assembly takes place in the host cell nucleus or in the cytoplasm. DNA virus assembled in the nucleus except the herpesvirus and poxvirus in the cytoplasm, and RNA virus gets assembled in the cytoplasm. From maturation we will continue in the next video.